Before I begin my talk this afternoon, I just wanted to say how great it is to be down here again. I've seen all my old friends and meeting many new ones, and I'm looking forward to doing the same over the next day or two. I want to start off also by giving a huge thanks to the Bahamas National Trust and College of Bahamas and all of the partners who made these meetings possible and brought us all here together. So thank you all very much. I'm talking today about spatial-temporal evaluation of somatic growth dynamics of West Atlantic hawkbills. Although I'm standing up here all by myself, there are actually 45 other people on this nice big stage sharing it with me. Two of my co-authors are in the audience, Alan Bolton and Stevie Connett. And this study was made possible by all of us joining forces, bringing together our relatively small data sets on hawksbill growth rates and combining them for the region so that we could answer much greater questions than any of us could have answered on our own. Okay. All of the authors would like, of course, to thank their many, many field assistants who have worked over decades helping them collect these data, as well as the funding sources. And Alan, Stevie, and I would like in particular to thank our Bahamian colleagues, the Bahamas National Trust, who are collaborators in all of our research in the Bahamas, Department of Marine Resources, who have allowed us over the years to have the great privilege of studying sea turtles in the Bahamas, Morton Bahamas Limited, the U.S. Coast Guard, and St. George's School, who've supplied invaluable logistic support. And most importantly, all of the good people of the Bahamas who have worked with us so wonderfully and cheerfully and efficiently and effectively catching green turtles for this study. Hawksbills, sorry, I'm getting my next paper's green turtles. These are hawksbills. All right. So hawksbills, fascinating species, very, very important keystone species on coral reefs. They're very closely tied to coral reefs, and they are a shared resource among many nations. This is truly a shared resource. Throughout the lifespan of an individual, they may well pass through the jurisdictions of 30 or more nations as they range throughout the greater Caribbean, as they grow slowly to sexual maturity, which takes 30 to 40 years for them to attain maturity. So I like to say that it takes an ocean basin to save sea turtles because the whole region has to work together to share these highly migratory animals. The U.S. National Research Council a few years ago called for sea turtle experts to work together to combine their data sets so that we could better answer some of the bigger questions that we have in terms of population assessments and conservation issues for sea turtles. And so all of us responded to that call by combining data from our 24 study sites, as shown here. And so we were able to cover a great region from Bermuda in the north to the islands off Brazil in the south and throughout the greater Caribbean, as shown here. We also have great temporal coverage, over 33 years worth of data from 1980 to 2013, and body sizes ranging from 19 centimeters, the size at which hawksbills first arrived into our benthic habitats after recruiting from oceanic habitats, up to 90 centimeters, which is the age at the size at sexual maturity. So for our analysis of growth dynamics, we used a generalized additive mixed models, and we used initially 10 potential covariates, 7 of which were fixed effects, 3 were random effects, and we tested for the effect of all of these covariates on growth rates. And our final model accounted for more than 42% of all of the variation in these growth rates, which was fairly impressive, given that there were nearly 4,000 growth increments in our data set. The key fixed effects that I want to talk about today are the 3 shown here. starting off with latitude and longitude surface, or the spatial, we were very surprised to find that there was no significant spatial effect throughout this entire region on growth rates of hawksbills. So hawksbills throughout this region were growing at very similar rates and were responding to the other significant covariates in the same way throughout the region. We found that there was a very significant effect on body size. This is nothing new. Almost every study of sea turtles has shown that there is a significant effect. Here we have this early, first of all, you're going to be seeing a few of these slides, so let me just explain it a bit. On the y-axis, there's something called a GAM response function, but actually just look at this as if this were annual growth rates in centimeters per year. And across the x-axis in this case is mean carapace length, the upper shell of the sea turtle, which is the most common measurement of size. So also the gray area are the 95% confidence interval around the point estimates, the dark line. So in red, you can see at the early part of the curve, we have slow growth rates, and we believe that's due to the fact that when these young hawksbills recruit from the oceanic where living was good and feeding on jellyfish was really easy, they have to adapt to feeding on a primarily sponge diet, which can be a great challenge enzymatically and physically to adjust to this new diet. And so we think that's this new adjustment part as their growth rates slowly increase. Then the growth rates decrease as they get larger, which is a general trend in sea turtles. There is this interesting little bump in the road, if you will, in the middle where I have the question marks. This is undoubtedly a significant shift, but we don't know what causes it, and we really need more research in this size range of sea turtles to figure out what is causing this shift in the dynamics. And then as the turtles increase in size and approach sexual maturity at around 70 centimeters, their growth slows down to almost nothing. The effective mean year was actually the most interesting covariate that we had. And you can see here that from 1997 on, there is a significant decline on growth rates of hawksbill turtles. And remember that because there is no change or no significant spatial effect or spatio-temporal interaction effect, these slowing growth rates are occurring throughout the entire region. And of course, this is of real interest. And so because we knew this was a regional effect, we thought first to look at some of the regional environmental indices that might indicate some relationship and explanation for these slowing growth rates. So in the top graph, we have plotted the mean annual growth rate. And in the middle plot, we have mean annual multivariate El Nino Southern Oscillation. And you can see there's a very tight correlation there of 0.99 between those two. Now, many of you may be surprised to hear me talking about El Nino here in the Atlantic, because that is, after all, a primarily Pacific parameter or indice. But in our readings, we came across a series of papers that made it very clear that there's a very strong teleconnection of this multivariate ENSO index with our entire region in the Atlantic. And so this is actually a rational comparison. And then on the bottom graph, we have a mean annual Caribbean sea surface temperature, which has a very high negative correlation of 0.85, negative indicating that as temperatures are increasing, growth rates are decreasing. Now, of course, these are correlations. They are relationships, not necessarily a cause of the change. And we can't rule out other possibilities, like perhaps density-dependent effects on growth rates. But knowing how severely over-harvested and overexploited these hawksbill populations were for so many hundreds of years, We doubt very much that at this point, although the populations are recovering in many areas, the recovery has been relatively small, and the populations are simply not at a size where there would be density dependence. Another line of evidence that suggests that this may well be a major regional environmental effect on productivity is when we compare our hawksbill data with those of our loggerheads, which showed a 37% decline in growth rates from 1997 to 2008, a similar decline in their nest numbers over the same time period as the hawksbills, suggesting again there is this regional environmental effect. And so this, of course, leads us to what about herbivorous green turtles? Loggerheads and hawksbills feed on the same trophic level, primarily on benthic invertebrates, but herbivorous hawksbills may have a very different pattern, and so right now we're gathering data for green turtles from throughout the same region, and we'll run a meta-analysis using those data. So what if temperature is the major effect on hawksbills? What could be the cause of that? And there really are two main avenues that we would consider. One is a direct metabolic effect. You know, that perhaps higher temperatures are causing higher metabolic rates, that feeding on their sponge diets, they can't process enough food to keep up with that increased demand. Or the higher temperatures may be exceeding an enzyme threshold in their biochemistry that is reducing growth rates. The other is through an indirect source, which is through their food resources on coral reefs. And of course, it's easy to say, well, we all know that coral reefs are in real trouble, particularly in the Caribbean. With the increasing temperatures, coral reefs are really suffering. And so, you know, the link to hawksbills would seem very clear. But it really isn't as obvious as that because as I said, hawksbills feed primarily on sponges in these coral reefs, and sponges are actually expected to do better under some of the models that are looking at increase in ocean temperatures. These are two species in particular that hawksbills really like, and these are suggested that these will do very well as temperatures increase, and that many of our coral reefs will become sponge reefs, so providing more food for hawksbills. So critical for us to continue to monitor growth trends over time in this species and many of our other wide-ranging regional species to try to ascertain how our ecosystem is going to be changing, and also to evaluate the effects of environmental parameters. Now in my last 2 minutes, I want to shift. I've just been informed by Ed here, who's tall and tough, and I don't want him throwing peanuts at me, that I have 2 minutes left. So I wanted to spend this on a new initiative that the Bahamas Sea Turtle Network is working on under the leadership of Indira Brown of the Department of Marine Resources. We are forming a network of sea turtle biologists and enthusiasts throughout the Bahamas. And our initial objectives are going to be trying to reduce the illegal harvest of sea turtles in the Bahamas that has unfortunately been increasing since the ban was put in place in 2009. And we're going to be taking these steps. This poster has been created that Indira Brown and Casuarina McKinney-Lampton have taken the lead in getting this poster produced, and these will be posted at appropriate spots to try to protect our sea turtle nesting beaches. And then also many education events going on throughout the Bahamas, led by Stevie Cannett and Barbara Crouchley, looking at classrooms. Stevie here has just asked his students, how many people have eaten sea turtles? And that's the hand raised. And then he next asked, how many have eaten sea turtles in the last week? And about half of the kids still put up their hands. So we do have some work to be done. So education, of course, is always the solution, and that's what we're working on. So thank you very much.